Hi everyone, welcome to our next lecture. Today we will be talking about eukaryotes. Just a couple of reminders here before we get started that eukaryotes include fungi, algae, protozoa, helminths, which are a parasitic worm, kind of like tapeworms or nematodes, including tapeworms and nematodes. And then we're also going to be looking at some microarthropods, which are small invertebrate animals with an exoskeleton and a segmented body that are visible to the human eye. But we also use microscopes for identification. Just some more reminders. Remember that infections caused by eukaryotes are more difficult to treat because our cells are also eukaryotic, meaning it's difficult to introduce a substance into the body that will harm only the eukaryotic cells that you want from the organism and not your own cells. Some other notes, just also always keep in mind that algal diseases are not infectious. They are intoxications, as the symptoms are due to the ingestion of toxins produced by the algae. It's not physically the algae, it's the toxins they produce that are making you ill. And then also want to just give you some background here about arthropods. Arthropods that transmit infectious disease are called vectors. These diseases are typically best controlled by decreasing the exposure to vectors, meaning if you keep humans away from these disease-transmitting animals, that's one of the most effective ways of suppressing the diseases that they can cause, or the diseases that they carry with them. So those are just a few notes before we get started. Just to orientate you guys, so up until recently, we've been dealing a lot with bacteria. So we've been on this left side of the 3-domain system. Now we're going to jump all the way over to the right side, looking at eukarya. So we're moving quite a ways away. That being said, if you look at fungi, which is one of the most frequently studied eukaryotic organisms in microbiology, you can notice that there are some differences in their characteristics that separate them from the bacteria that we've been looking at so far. So first of all, their study of fungi is called mycology, and they are of the domain Eukarya, kingdom of fungi, and their metabolism is they are chemoorganotrophs, and they can be both aerobic or facultatively anaerobic. Just as a note here, many fungi rely on saprophytic or lysotrophic nutrition. So lysotrophy is a process of chemoheterotrophic extracellular digestion involved in the processing of the decayed organic matter. Their cell types, as we very much expect at this point, they're eukaryotic. Some very important differences that I want to point in here is note in the cell wall the lack of peptidoglycan, and that they have chitins in their cell wall. This is very, very characteristic of fungi. And I'm going to let you do the rest of those characteristics, save for I will mention that we will be discussing both the sexual and asexual spores and reproduction processes of fungi. So moving on to the vegetative structures of fungi. So first of all, you should be aware that the thallus is what the body of the fungus or the mold is called. And then the thallus is held together by long filaments or cells that are joined together, and these are the hyphae. And these hyphae can be filaments with or without septa, or septa is also just another word for cross walls. So these filaments can either be long, one continuous, or they can be little, it almost looks like they're a little bit segmented. We'll see an example of that in the next slide. Most molds have septa that divides the hyphae into distinct uninucleate cell-like units. Note the uninucleate here, meaning all of these little distinct units have their own little nucleus. Then the exceptions, of course, as we are aware, many of these microbes have exceptions to the rules. There are a few exceptions here that lack septa. They're also called co-enoxytic, and thus the hyphae appear as long continuous cells with many nuclei. So it's literally just the difference of whether the walls are present or not, and if the walls are present, they will separate these little segments into only having one nucleus, and if the walls are absent, you'll have this elongated cell that contains many nuclei. Hyphae grow by elongation at the tip. We'll see an example of that also in the next slide. And it's also notable that each part of a hyphae can grow. So when a fragment breaks off, it can form new hyphae. And in the lab, actually, typically fungi are grown from fragments obtained from the fungal thallus. So you will take a small segment and then you will just have it grow on its own, and that is how you will grow them in the lab. Just a note here also that fungal colonies are called vegetative structures as they are composed of cells involved in catabolism. Remember, catabolism is the breakdown of molecules and growth. So, sorry. One too many. There we go. Okay, so here's our pictures. Remember I said in the previous slide that we will be looking at the hyphae that have cell walls. Here you can see those. So you can see there's your septum, your cross walls, and how they separate your hyphae into these little segmented pieces that all have one nucleus. Now on the other side, here you have a filament that lacks septa, so you'll see it's one continuous thing lacking all the cross walls with many nuclei contained inside. And then the picture on the right, which we see over here, is just the growth of a hyphae. Here you can see the spore and the hyphae starting to form, and note how it just elongates at the tip and it just keeps growing in that direction. So moving on to mycelia or mycelium. So mycelium is a mat of fungi that is visible to the naked eye. It's essentially just a mass of hyphae. And they are separated into two types, vegetative or reproductive. So you can think of vegetative hyphae as non-reproductive, as their primary function is obtaining nutrients. And then the counterpart is, of course, reproductive or aerial hyphae. And here on our picture here, you can see the difference. So here, the one that extends into the air would be your aerial hyphae, or your reproductive hyphae. And I would like you guys to take a few seconds to think why this would be the case. Why is this extending vertically upwards? And the answer to that is it's an evolutionary trait where this it helps the organism to spread its pores. Because if the spores are released down here with all these other hyphae and filaments in the way, it wouldn't be able to distribute their spores so effectively because it relies on air movement to distribute these spores. So getting your reproductive organs into the air, or your reproductive organs is not the right word, your reproductive cells into the air gives you that advantage of being able to distribute your spores more effectively via wind. And then at the bottom here, you can see your subsurface or your vegetative hyphae. Okay. Moving on to molds, just some examples here, and I'll show you the difference between the aerial and the vegetative hyphae on the pictures again. Just as a reminder, remember that the fungal thallus consists of these hyphae, and a mass of the hyphae is called a mycelium. There are your differences between what a thallus is and what a mycelium is. Now, here you can see on the picture your vegetative hyphae, which are these strands that link the bodies together. And here you can see your vegetative hyphae. Same here, your aerial hyphae is more towards the center of your mold, and your vegetative hyphae is essentially your nutrient gathering hyphae are spread out here on the side. So now we're going to be looking at yeasts. So what's the difference between a yeast and a mold? So typically yeasts are non-filamentous unicellular fungi that are typically spherical or oval. Very important to note that they are non-filamentous. They don't produce the filaments. And they are frequently found, as an example, just to give you guys an idea of what we're talking about here, as that white powdery substance or coating that you can sometimes find on fruits or leaves. That would be a yeast that's growing there. So in this picture that we have over here, this is a picture of yeast cells in young wine, and here you can note their spherical and oval shapes that is so characteristic of these cells. I would also like to note to you guys that these organisms typically reproduce by budding, and you can where one organism or one cell can eventually form up to 24 daughter cells. So yeah, that's something that's a little different to them than the bacteria that we've been working with so far, or the typical bacteria that we've been working with so far. Also, something important to note about these organisms is that they can be dimorphic, or they can show dimorphism. So what is dimorphism? Di, as you guys are hopefully aware, means two. So dimorphism means there are two morphologies. So that means that they have two forms of growth. These fungi can either grow as a mold or as a yeast. The mold-like forms produce vegetative and aerial hyphae, as we just saw in the previous slides, whereas the yeast-like forms reproduce by budding. So something that oftentimes drives the mechanism that the fungi will use is temperature. So with pathogenic fungi, for example, they're typically yeast-like at 37 degrees Celsius. So in our, the human body temperature. And they're typically mold-like at 25 degrees Celsius, which is commonly accepted as room temperature. So in that 12-degree difference there will change the fungi into either appearing as a yeast or appearing as a mold. So here I would like to also, with the yeasts, that they can use fission or budding to reproduce. And in fission, it's the cell will divide symmetrically. So an example of that will be here on the left. Look at how symmetrical these two cells are, and you can see how they're getting split exactly more or less in the middle. So they look very, very similar. Where budding, which we've seen an example of on the right here, divides the yeasts asymmetrically. Look at how much smaller the bud is than the parental cell. And they also leave budding scars or bud scars, because you can see this cell will eventually separate, and then this is the scarring that it will leave over on the parental cell. So this is a nice picture to show you the differences between fission and budding. Here is our example of dimorphism, or a picture thereof at least. This is of Mucor indicus. And you can see that it has both the yeast-like growth. Look at those spherical or oval shaped cells over there. And there's also the mold-like growth in between that. Look at these hyphae and these filaments, or the filamentous growth. And again, note that there's that temperature difference that drives whether it will grow as a yeast or whether it will grow as a mold. So now moving on to fungal reproduction. So fungal can produce asexually, most common, or they can reproduce sexually, very important there. So how would it do asexual reproduction? So that's simply through the formation of spores. I cut this picture from the textbook that you guys all hopefully have at this point. And it just shows you how these asexual spores are produced. So they are produced via mitosis and cell division and formed by the hyphae of the organism. And the terminology here is a conidiospore is unicellular, or a conidium is multicellular. So here you can see, starting here at number 1, that the aerial hyphae produces sporangium. This is the structure that holds your spores. The sporangium bursts open to release your spores. So you can see your sporangiospores being sprayed by the wind or by the air. And the spore will then land somewhere and germinate to form a hyphae. And then this vegetative mycelium will start growing, and the process will repeat. And that is how your asexual reproduction will take place. What I do want you guys to note here is I purposefully left in this little segment, because this is the other half. This is the sexual reproduction half. And notice the numbering when we get there. It starts immediately at 5. So the sexual reproduction, it can convert from asexual to becoming sexual, and then vice versa. So the cycles can be interlinked. It doesn't necessitate that if a fungi is using asexual reproduction, that it has to stay that way. It's important for you guys to be aware of that. So if you move on here, again, notice the numbering, how we start immediately at number 5. So this is after your spore has landed somewhere and the mycelium has grown. Now there will be gametes that form at the tip of the hypha. Then plasmogamy will happen. So what is plasmogamy? Plasmogamy is a stage in the sexual reproduction of fungi in which the protoplasm of the two parent cells, usually from the mycelia, fuse together without the fusion of nuclei. So they're effectively just bringing two haploid nuclei close together in the same cell. In the zygospore forms, what is the zygospore? The zygospore is a thick-walled resting cell that certain fungi and algae have that arises from the fusion of two similar gametes. Then karyogamy and meiosis happens. So what is karyogamy? It's the final step in the process of fusing together two haploid eukaryotic cells and refers specifically to the fusion of the two nuclei. So up to this point, the nuclei were close together, but they were separate. Now the nuclei are being fused together. And then before this process, each haploid cell contains one complete copy of the organism's genome. And then again, this should look familiar because this is the same process that this asexual reproduction followed. So the zygote will then produce a sporangium. The sporangium will burst and the spores will be released. The spores will germinate and a hyphae will be produced, and then the mycelium will start growing in. But notice again that you can then, from this point on, you could move back to asexual reproduction and vice versa. Okay, so just some extra notes here that I want you to be aware about regarding the feeding and some of the behaviors and pathogenicity of fungi. So fungi typically feed by the use of extracellular enzymes such as cellulase and pectinase. So essentially they secrete these substances that then break down compounds, and then they will absorb the nutrients from the broken down compounds. They're specifically very well adapted to breaking down recalcitrant compounds, and a recalcitrant compound simply refers to a compound or organic material that is very, very resistant to decomposition. Think about when these fungi will feed. So typically fungi, saprophytes, will be decaying organic matter that's already dead. So this is after the other organisms, I mean, if you think of an example of an antelope, for example, dying and a lion eating it, the most easy to break down and easy to get to nutrients will be broken down by your first microbes that arrive on the scene. But fungi arrive much, much later and are left with the compounds that other organisms typically cannot break down and can't feed on. So they have to be very effective at breaking down and extracting energy from these resistant compounds. So then there are also a few pathogenic fungi that are both pathogenic for humans and animals, but there are way more that are pathogenic to plants. Plants are typically the ones that are hit hard by fungi. So if you work in agriculture or forestry, you will definitely come across fungal control and how to mitigate the problems about pathogenic fungi regarding plants. Okay, and some nutritional adaptations for fungi. So typically they grow based on a pH of more or less 5. So this is acidic conditions that they like to grow in. And they can also grow in high sugar and salt concentrations. So they are resistant to osmotic pressure. They aren't as negatively affected by that as many other microbes are. They also capable of growth in low moisture content. And they can metabolize complex carbohydrates. Again, think of when they feed. Something that is also important for anyone interested in the plant side of things is mycorrhiza. So what is a mycorrhiza? A mycorrhiza is a mutual symbiotic association. It's also potentially mildly pathogenic, but that is maybe a little still unclear or a little disputed at this point, but I just thought I'd mention it, between a fungus and a plant. So the term mycorrhiza refers specifically to the role of the fungus in the plant's rhizosphere. So the rhizosphere of a plant is its root system. The mycorrhiza play important roles in plant nutrition, soil biology, and soil chemistry. So essentially what happens is the mycorrhizal fungi will colonize the root system. So here you can see this over here is a root hair. So this is what we're looking at, a very, very, very small structure here. This is a root hair, and then on the pink here, you have an arbuscular mycorrhiza, and the blue on the other side, you will have an ectomycorrhiza. So these are the fungi that will then penetrate the root hairs. So what this does is the fungi will produce, will improve the plant's capability of uptaking water and nutrients such as nitrogen or phosphorus, while the plant will provide the fungus with carbohydrates that it gains from its photosynthesis. So that's why it's considered a symbiotic relationship. And more than 90% of all plant species will benefit from this type of symbiotic relationship, and it is associated with many, many plants, again, also over 90%. And then the difference here that I want you guys to be aware of is arbuscular mycorrhiza. These fungi will penetrate your root cells. So these root cells are represented by these white blocks here, and you can see how the fungi is actually going through them and penetrating into them. Whereas your ectomycorrhiza your fungi will surround the cells, but they do not penetrate them. So that's the difference between the two of them. And then going on to maybe the most commonly thought of fungi, saprophytic fungi. So sapro comes from the Greek word meaning rotten, and they decompose dead organic material like fallen trees, leaves, and soil organic matter. And they are predominantly responsible for the decomposition of recalcitrant organic material. We've discussed this before, so I'm not going to spend too much time on it, but that's really, really, really important. Because that is a role that these organisms can fill that many other microbes are unable to do. Now we're going to be discussing lichens. Here you can see a nice little picture of a lichen. This is a lichen that grows on a tree, obviously, but they can also grow on rocks very, very commonly. So what is a lichen? A lichen is a mutualistic or a symbiotic combination of a green algae or cyanobacterium and a fungus. So in this association, the alga will provide carbohydrates for the fungus. So this is similar to the mycorrhizal example where the plant provided carbohydrates for the fungus. And then the fungus will provide protection, water, and nutrients for the alga. So again, you see this water and the nutrient provision that stems from the fungus. This is again the same as it was in the mycorrhizal relationship. So these lichens will produce acid that can weather rocks, and then they can absorb some of the nutrients from the rocks. So what type of heterotrophs would they be, or chemotrophs, or where would they fall on that spectrum? You guys should know this by now. And then there's these organisms typically also have a long lifespan, and they have a slow and regular growth rate. Interestingly enough, back home, we used in some of the geomorphology courses, we used lichens growing on gravestones to estimate their growth rate and their, because I mean, the gravestone obviously has a date on it of more or less, it gives you an idea of when the stone was there. So you know the maximum age of the lichen. So that's an interesting way that we can study lichens. I mean, some of those graves go back, many, many, many years. So yeah, that's an interesting way that you can study lichens. And just another fun fact about lichens is that lichens are actually found on about 6 to 8% of the planet's surface. So they are, very, very numerous. Now we're going to be moving on to algae. Important here that algae are not a taxonomic or a monophyletic group. Algae is simply a way that we describe phototrophs that lack the tissues to be classified as plants, meaning they don't have roots, stems, or leaves. So these are that typically includes your unicellular or filamentous photoautotrophs. Again, they lack roots, stems, and leaves, and they are mostly aquatic, as water is required for their growth and reproduction. And here gives this table, I decided to include it just because it gives you a nice idea of some algal diversity, um, regarding their colors, their cell walls, and their cell arrangements, their photosynthetic pigments. This is a big one for algae. The sexual reproduction, whether they have sexual reproduction or not, which many do, as you can see, very important. There's storage material and the pathogenicity. Remember again here that algae typically produce toxins. So they are, it's the toxins that make you ill that the organism produces. It's not the same as when a bacteria is attacking your cells. So something that I thought was also always very, very nice to look at. I definitely advise you guys to maybe Google a few pictures of these things. They're called diatoms. So diatoms are single-celled algae which have a cell wall of silica, and they are typically planktonic organisms, meaning they're free-floating. So they are unable to propel themselves against the current, so they just kind of float around in water. And they are so, so pretty to look at, as you can see over here. And something that's different about these microbes to others is scientists have found extensive fossil deposits of diatoms. That's something that you don't typically find from microorganisms, of course. So that's definitely something that's very interesting about diatoms. Something that I do want to explain to you guys a bit about, and I'm not sure how much of this is in your textbook. I did find some of it, but I think I'm going to expand on it a little bit here, and going a bit more into the microbial ecology and ecology sides of things. So if you look at algal habitats, this example that we have here, you will find that you have green algae near the surface, and they absorb red and blue light. And then you will find red algae at the bottom because they absorb only blue light. So the blue light penetrates further down than the red light due to the wavelength. The longer wavelength doesn't allow red light to penetrate so deep into the water. And this is a very nice example that you can use to explain what an ecological niche is and the differences between a fundamental and a realized niche. So what you can deduce from this is because the green algae are able to absorb both red and blue light, but the red algae can only absorb blue light. So if the red algae were absent, you would expect to find green algae throughout this entire, depth range, meaning its fundamental niche of the green algae is the entire, this entire depth range here. However, once in these areas where the blue light is the only light that is present, it is outcompeted by the red algae. The red algae are more efficient at growing in these areas than the green algae are. So they, thus, when you move down to this depth, you only find the red algae and no longer find the green algae. And this is the difference between a fundamental and a realized niche. So the realized niche of the green algae is just this top portion here. But its fundamental niche, the niche that it could theoretically occupy, is the entire depth. But it cannot occupy that because it gets outcompeted by the red algae at the greater depths. Just something that I thought would be interesting for you guys. I know WVU, I think Dr. Morrissey typically presents a course on environmental microbiology, where you will then learn more about this. I definitely recommend you guys take it. It's a pretty interesting course. I took it here a couple of years ago. And that's where you learn concepts of, some ecological concepts about fundamental niches and realized niches and the like. So it's really, really interesting. Now, the relevance of algae. Why do we care about algae? So typically, the first starting point would, of course, be that algae serve as the basis of the marine food web. So here you can see these microorganisms, and the algae will also form part of this at the bottom of your food web that feeds the, your bottom, your organisms lower down in the food chain, which then will feed the higher up and the higher up. So a disruption to these organisms, or a change or a decrease in them, for example, could disrupt your entire food chain, just because there would be no food. They also produce at least half of the Earth's oxygen, so it's sort of important if we want to breathe. And they're also capable of causing harmful algal blooms. This is what you can see over here. This green, it almost looks like that's melting into the water, but this is actually algae. And this algae is typically grows because there's an excess of nutrients, also known as eutrophication, where this excess of nutrients just cause massive amounts of algae to grow. And initially you would think, okay, well, that's not so bad. But the problem is that these algae can release toxins, so they can be toxic. And then they can also lead to dead zones because they can block sunlight from reaching other organisms. They can deplete oxygen levels in the water. So yeah, they can really create a massive problem. There are multiple examples of that. I mean, you can look at the Gulf of Mexico has a big problem with algal blooms, and many places, because it's typically agricultural practices where we use a lot of fertilizers that then get washed out with rainfall and surface runoff events, and these nutrients get transported to water that then gets transported to stationary water. All these streams coming together will eventually reach a lake, where then you can have these algal blooms, or they can even wash out into the ocean, leading to also algal blooms in marine environments. So just a note for you guys here. Please be sure to have a look at algal reproduction. I'm not going to be discussing it today, just just due to the time. I do want you guys to be aware of both the sexual and asexual reproduction of algae for the upcoming tests and exams. I don't think there's anything too insane. So yeah, just be sure that you have a look at that. Now we're going to be moving on to protozoa. So what are protozoa? These are pretty interesting microbes, in my view at least. So the term proto means first, or like you almost think of it as almost pre, but proto, think prototype, it's like your first one. It means first animal. So protozoan, zoa, referring to animals, protozoan then being the first animal, and it typically describes its animal-like nutrition. So these unicellular eukaryotes inhabit water and soil, and their life cycles typically includes reproduction by fission, budding, and schizogony. There we go, schizogony. So that is essentially multiple fission, meaning the nucleus undergoes multiple divisions prior to cell division. So the nucleus divides numerous times before the cell actually splits up. So after these multiple nuclei are formed, a small portion of the cytoplasm concentrates around each nucleus, and then the cell separates into daughter cells. Just as a note here, that sexual reproduction has also been observed in protozoa, and these organisms are also capable of encystment, which means that they form cysts under adverse conditions. And cysts in this case is essentially just a protective capsule that they can form around themselves, and they typically require a large supply of water. So protozoa also have this adaptation that they have a pellicle surrounding the cells, an outer protective pellicle, and thus they require specialized structures to take in food. So if you look at ciliates, for example, you can see an example of one here. They have these cilia on their and that grows on the outside of the pellicle, and essentially these cilia can wave food towards the mouth like cytosome. Here you can see the cytosome. So you can almost think of them as little fingers that help guide food into where they need to get the food to eat them. And then they will use these little food vacuoles. So essentially, once the food enters this cytosome, you'll be included. I can almost think of a little inclusion being made, and then they will use these vacuoles or food vacuoles to digest their food, and then they will eliminate waste through an anal pore. So there are definite similarities there between animals, higher order animals, and these protozoa and their digestive systems. Or you can see how this could be a precursor to something greater, I suppose, is a better way of phrasing it. The engine also, as a note, is that amoeba phagocytize food, so they just envelop the entire thing. That is how they will eat food and digest their food. Something that I also want you guys to know here is what a contractile vacuole is, or CV. It's a specifically subcellular structure or organelle that is evolved in osmoregulation. So what does osmoregulation mean? You guys, I hope by the structure of the word you know what it is, and it is found predominantly in protists and in unicellular algae. And osmoregulation refers, of course, to osmotic pressures. So the moving on to some amoeba, just to talk about them here for a few minutes. So they move by extending pseudopods. I definitely would advise you guys to go look it up. It looks pretty weird. It looks like this little organism just grows feet to move around with. It's really pretty cool. And then they're also medically important. So if you look at something like Entamoeba histolica, they cause amoebic dysentery. And this is the only pathogenic amoeba that is found in the human intestine. And up to 10% of humans may be colonized by this amoeba. Then there is also amoeba that leads to the infections of corneas that can cause blindness. Palamuthia, which is a granulous amphibic that causes granulomatous amoebic encephalitis. This encephalitis is inflammation of the brain. Something that I also want you guys to know is that slime molds, that I'm sure many of you have seen it. There are quite a few videos going around of these yellow molds that they use to solve mazes. It's pretty interesting. And they're also closely related to amoeba, although not exactly the same thing. Definitely worth looking up what slime molds are and reading those sections in your books. Then something else is these Apicomplexa, which is a phylum of parasitic protozoa that are the one characteristic about them that's really stands out, the fact that they are completely non-motile and that they are obligate intracellular parasites that have very complex life cycles. So an example of this would be Plasmodium, which causes malaria, big problem where I'm from. It's definitely worth checking out the YouTube video and reading up about their life cycle. It's definitely different because these organisms can only reproduce once they are within the cell that they are parasiting. So that's without those cells, they can't. You can almost think of them in that sense. They are similar to viruses, where if they're out of the cell, they can't do anything. But once they are in the cell, then they can actually reproduce and cause some serious harm. We're also going to look quickly at helminths. So helminths are multicellular eukaryotic animals, and there are two main phyla. There are the platyhelminthes, which are flatworms, and the nematoda, which are roundworms. And some of these are parasites that are specialized to live in hosts. Interestingly enough, many of them lack a digestive system. They have a very reduced nervous system. They have limited or reduced locomotion, or they might even lack locomotion. But they do have complex reproductive systems. So this is an example of cestoda, which is tapeworms. I mean, dear God. Remember at the start of the semester when I told you many of these things look like little aliens? It's like studying alien life. Yeah, I mean, that looks like something straight out of some sci-fi horror thing. And just important about these platyhelminthes is that these flatworms are dorsoventrally flattened. Moving on to nematoda, so these are very important in soils. They are soil inhabiting nematodes. They help accelerate decomposition processes, and they are very beneficial for the recycling of nutrients in soil. And they can often feed on bacteria and fungi. There's another video you guys can check out about nematodes. And then finally, we're going to be looking at microarthropods. So these are microscopic invertebrates. Many of them live in soil. So there can be parasites included there too. There's an example here of a dust mite. There's another little video that you can check out about microarthropods. And then I just want to take a minute or two to just talk about the soil microbial food web, and sort of bind everything we've been talking about together, looking at the different trophic levels and how everything is essentially interrelated. So you can see here that once the plants die and leave behind some organic matter, the soil food web is really stimulated by this organic matter, because this is often then eaten either by fungi or bacteria. And then if it's bacteria, it can be then these bacteria can then be eaten by protozoa, which would then be your third trophic level. And the protozoa can then be eaten by nematodes, your fourth trophic level, which are typically eaten by arthropods. And then arthropods are actually large enough that they can be eaten by small animals and birds. From the other side of things, if it's fungi, you can go from the fungi immediately to nematodes and arthropods. And then from the arthropods, they can also be eaten by larger arthropods. And again, then you're at that level where they can actually serve as food sources for either birds or small animals. But you can also see just the important location of fungi on this food web, or in this in the soil microbiome, if you will. If these organisms were to go missing, it would disrupt the food web, where you would struggle to get rid of organic matter. It might result in decreases of your arthropods and nematodes, which could lead potentially to increases in bacteria, which can include harmful bacteria. But yeah, so just long story short, if you remove these microscopic eukaryotes, you would disrupt your entire soil food chain, and this would disrupt nutrient cycling, and it would disrupt just how nutrients move throughout the environment and become useful to us and to other organisms. Okay, then for today, we're also going to do 4 review questions. So which of the following is not eukaryotic? Fungi, algae, protozoa, helminths, or archaea? I mean, this is a very, very easy question. I'm just going to give you guys about 10 seconds, or not even like 3 more seconds. The answer, of course, here is archaea. I mean, it's literally a different domain. So I'm hoping everyone got that one correct. True or false? Lichens exclusively comprise of fungi and algae. So what are lichens made of? What are the 2 organisms that form a lichen? So the answer here is false. Remember that instead of an algae, you could also have a cyanobacteria. You just need a photosynthetic partner. You need a fungi and a photosynthetic partner for it to be a lichen. Again, I've said this to you guys many times. Be very careful when making generalized statements. You have to be very, very sure that there are no exceptions and nothing that you've overlooked before you make statements such as these. So, true or false? Arbuscular mycorrhiza penetrate root cells. So remember, think back that there are 2 types of mycorrhiza. So was it the arbuscular that penetrated root cells, or was it the other one, the ectomycorrhiza? The answer, of course, here is true. It is actually the arbuscular mycorrhiza. And then our final question for the day. Fungi can metabolize complex carbohydrates. Is this true or false? I'm going to give you guys about 5 seconds. And the answer in this case is, of course, true. Think, again, really important, think of when fungi would feed. If there are saprophytes, specifically saprophytic fungi. And then these complex and recalcitrant organic matter will be all that is left by the time that these fungi are able to, feed. So yeah, they can definitely metabolize complex carbohydrates. It's one of their distinguishing features. Okay, guys, that'll do it for today. I hope you guys are doing well, and then I'll see you guys in the next one. Okay, cheers.